we will start discussing about the problems that arises with the immune system. Till now you have learned about what the immune system is, what are the parts of the immune system, and how this immune system protects the human body from various diseases caused by microbial agents. Now, basically, if we do divide the immune system, we have the innate and the acquired immune system. The acquired immune system adapts as the human body develops and grows. Now, with the immune system, some kind of problems may arise. The first problem is if the immune system fails to act the way it should, either because some of the cells, some of the proteins, some of the important enzymes are missing, then we call it as immune deficiency diseases. Similarly, on the other hand, various scenarios can arise when the immune system does functions. Either it functions too vigorously so that it actually harms the human body more than what it is intended to do, and sometimes it may actually directly harm body's own cells. That is what we call as autoimmune status. That means the body starts reacting to the immune system against the own cells. Now, if we look back, a typical description of the immune system is shown like the armed forces which protects the country. Now, sometimes the armed forces, if it is deficient based on the country's geographical or population, we can call it to be deficient. Similarly, if the forces they are not acting like it is required to do, or acting rather irregularly or funnily, we call it as a dysregulated immune system. Among the important dysregulation comes the autoimmune disease and the hypersensitive disease. In today's class, we will look into what are autoimmune diseases and how and why it does occurs. If you do recall, the immune system acts in two ways: by directly acting on the cell and by producing what are known as antibodies. Now, first of all, I think you have learned about the major histocompatibility complex. Yes or no? Yeah. So, whenever an antigen does enters into the body, this antigen is processed by antigen presenting cells. Okay. This antigen may be foreign or may be self. Now, the antigens, some antigens are expressed in all nucleated cells of the body. They are recognized by MHC class 1, which activates and triggers what are known as CD8 positive cells, also known as killers or cytotoxic cells. What actually happens is that these cells scan all nucleated cells of the body, and if any nucleated cell of the body fails to express the MHC properly, these CD8 cells will kill that cell. Okay. On the other hand, if we look at the MHC class 2, that is expressed by all antigen presenting cells. That means this would include all the phagocytes, the B cells, and even dendritic cells that are present on the skin. Now, what they do? They will take up the antigen and express and will lead to triggering of the CD4 cells. Okay. Now, this you have learned. Now, let us try to understand how exactly occurs. In the nucleated cells, the proteins are there in the cytoplasm itself. These proteins are cleaved by proteasome, taken up by the endoplasmic reticulum, and then expressed on the cell wall. These proteins are then recognized by the MHC. If the cell had any kind of alteration in the protein expression, be it due to a viral infection which leads to a altered gene expression, or it could be even expression of proto-oncogenes which may lead to tumor genesis. If these proteins are expressed, then the CD8 cells will recognize them and kill. But if we look at the MHC class 2 pattern, the antigen presenting cells take up the proteins that are present extracellularly, not the intracellular, but the extracellular proteins. Then they express on the MHC class 2. This will lead to triggering of the CD4 cells. Now, what would be the action of the CD4 cells? Predominant action would be it would start producing cytokines which will involve the B cells, so that this particular antigen is processed by the B cell and antibody against that particular antigen can be produced. Okay. Now, why I am telling you all these things? Because when you understand how the antigen presentation occurs, you must understand that there are two things that should be present. One is an antigen or a protein that is present extracellularly, and a antigen presenting cell. Commonly, all the antigen presenting cell forms the part of the innate immune system, right? So, antigen when triggered along with innate immune system will lead to production of antibodies. This is the same principle that is followed when we are trying to produce antibodies iatrogenically, that is when we are trying to give a vaccine. If we only give the protein to the human body will not process that protein. We need to trigger the innate immune system. That is the reason why in all the vaccines you will have some kind of adjuvants. What these adjuvants do? They trigger the immune system. It could be a dendritic cell in the skin, it could be a B cell, it could be a phagocyte. Okay. So, this is all about the gist of how our immune system protects by the cell mediated and the humoral immunity in short. But then the question lies, then why we are discussing autoimmune diseases over here? Let us look at the definition. What the definition says? It's a condition in which body's own immunologically competent cells or antibodies act against the cell antigens. Please keep in mind it is immune competent cells or antibodies. That means both cell mediated and humoral immunity. Both can act against body's own cells. One of the earlier concepts was given by Paul Ehrlich, but let us consider the fact that this immune system has developed or evolved along with the human beings. That means in a normal mechanism there must be a mechanism by which the immune system can differentiate what is self and what is non-self. So, this differentiation can be understood by the concept of tolerance. Tolerance means the immune system tolerates some of the proteins, that is the antigens, which are of self origin. That means the proteins that constitute human body's own cells. This is called as tolerance. However, it does not tolerate any foreign protein or foreign antigens. This tolerance activity can act at two levels. One is called as the central level and the peripheral level. Now, what is this central and peripheral? If you do recall, why the T cells are called T cells? Because they are developed in the thymus, and B cells in the bone marrow, right? So, where they originate, if the tolerance activity occurs over there, we call it as central tolerance, and if after the cells has developed, then some activity occurs for the tolerance, we call it as peripheral tolerance. Okay. Now, briefly we can divide them as central and peripheral tolerance, because central means at the origin and peripheral means at the peripheral tissue. How it can occur? Look at this diagram. Now, if there is a weak or no recognition of self antigen, those cells will undergo apoptosis. If there is strong recognition of cells, it will be either apoptosis, that means that lineage of cells will die, or there will be a change in the receptors. That means the receptors which are recognizing the self antigen will be altered, or the development of those cells will be hindered upon. And if nothing that sort occurs, or if there is weak recognition, then only that cell will move from the central region to the periphery. Similarly, in the peripheral tissues, what will happen? Even then, if it recognizes self antigen, it will undergo apoptosis or anergy. What is anergy? Anergy is failure to act upon when required. That means a normal cell will act to produce either cytotoxic activity or production of antibodies. But these cells, because it does recognize a self antigen in the peripheral tissue, it will fail to act. That means if it is a B cell, even if it gets exposure repeatedly to the same antigen, it will not produce antibodies. Okay. So anergy means a cell which fails to act what it should. And finally, some of the receptors of cytokines will lead to suppression of the activity of this cell. This is how the central and peripheral tolerance occurs in gist. If we look at it slightly more, what will happen in thymus is that in T cells developing, these thymic antigen cells will process with self MHC, and all the cells which does recognizes the self MHC will undergo negative selection. That means all cells which recognizes the self antigen will die. Similarly, in the bone marrow, there will be receptor editing also. What is receptor editing? The B cells will have some receptors which can identify the antigens. Now, if these antigens do recognize self antigen, those receptors will be modified. Receptors are protein, so protein expression will change and those receptors will change. So that two things might happen. Either it will fail to recognize the self antigen, or even if it does recognize the self antigen, the feedback or the next step will not be processed. And like T cells, negative selection also does occur. That means all the cell lineages of B cells which do recognize self antigen will ultimately die off. Two important negative inhibitors in the T cell lineages are CTLA4 and PD1. The first one, major site of action, is in the secondary lymphoid organ, that is the lymph node, and the PD1 acts on the peripheral tissues. Now, why I am telling you these two? Not recently, many years back, this was discovered, and this is one of the very important steps by which the T cell activity is inhibited. When I say inhibited, that means a cell expressing these will not be killed by the cytotoxic T cells. Some of the tumors, they express these kinds of cells. Hence, they are not killed by the tumor cells. So, it was discovered in such a way, but now the action of these cells have been more, people have learned more about them and more expression has been done. Important part is that CTLA4 prevents autoimmune responses to self antigens, and PD1 prevents excessive responses to microbes. Why just keep this in mind? I'll talk about molecular mimicry in a little later part. Now, coming to the B cells, in the central tolerance part, we have two types of cell or antigen recognition. First type is a high avidity and other part is the low avidity. I think you have learned about avidity. Avidity means the ability to bind to that particular antigen in a degree of specificity and So, in high avidity, if there is non-self reactive B cell, this will undergo development, whereas if it does recognize the cell, it will undergo deletion, that is negative selection. But if the avidity is low, it will become anergy, that means it will not produce antibodies. So, if we do summarize what are the methods of peripheral tolerance, first would be ignorance. Some of the T cells, the self-reactive T cells might not ever encounter self-antigen when they recognize it. Because it has not been processed, it cannot even recognize them. Second would be anergy, I have already told you. Third would be phenotypic skewing. What is phenotypic skewing? Self-reactive T cells interacting with antigen presenting cell which present self-antigens undergo full activation. However, non-pathogenic cytokines and chemokines will not activate the cells. Second would be apoptosis. This is mediated by activation induced cell death. The first ligands will be activated and will cause apoptosis. Similarly, another type of cell is called the T regulatory cells. These type of cells can down-regulate the cells. It's a control mechanism within the T cell immune system. And dendritic cells, immature or tolerant dendritic cells will not process the self antigens. Another important part is sequestration of self antigen. In anatomy classes, we have learned about some of the proteins that are sequestrated in the human body, like the corneal protein, lens protein in the eye, and spermatozoa. They are not exposed to the immune system. Hence, human body cannot recognize these antigens. Then, having understood this, let us try to understand then how does the autoimmune status does occurs. That is how the human immune system gets triggered against the self antigen. This is the composite diagram, take a look at it. First would be a genetic susceptibility. That means because of some errors or some permutations or combination of the genes, this negative selection of self-recognizing cells may not be totally complete. On the other hand, we can have tissue injury and inflammation because of any trauma or infections where self-antigens gets processed and expressed. Furthermore, these self-antigens then will cause autoimmunity, or what I have earlier said is a molecular mimicry. What happens is that when a microbial agent is being destroyed by the immune system, various types of proteins are expressed as antigens. Some of these antigens or proteins may be similar to the human proteins. Now, when these proteins are expressed, antibodies or cytotoxic activity is triggered. Now, these cells then cannot differentiate between the protein of microbial origin and the similar protein of human origin. This is called as molecular mimicry. Okay. So, first would be breakdown of T cell anergy. Anergy, I have told you, if some cells do recognize self antigen, they will undergo anergy. That means they will not give the positive feedback to the cascade of the immune system. This is mediated by the B cell. And if it fails, what will happen? The loop will be completed. If apoptosis fails, that means cells which recognizes self antigen are not killed, they will remain in the system and start producing antibodies or cytotoxicity. If the regulatory cells fail to act, then we can have that scenario. Sometimes the T cell may process and trigger the B cells to produce antibodies. Then, or if there is release of sequestrated antigens. How this can happen? I have already told you. For example, the lens protein is a sequestrated protein. That means human body has never been exposed to this protein. Suppose if a person does get a trauma to the eye and there is injury to the lens and the lens protein is now exposed to the immune system, the immune system will start reacting against that lens protein, and reaction would lead to effect on the other eye, that is the uninjured eye. Why this happens? Because during the development of the immune system, these proteins have been anatomically isolated. Another would be a polyclonal lymphocyte activation. Some of the infection may trigger activation of many clones of cells, and when this is triggered, many clones of cells will start acting together, and when this vigorous reaction occurs, these regulatory pathways and then apoptosis or cell death, regulatory T cells, their activity will not be sufficient enough to control the reaction. Another would be epitopic spreading. What is epitopic spreading? In these, the cell peptides which are released due to persistent inflammation induced due to tissue damage are processed and presented by antigen presenting cells. There may be a shift or spread of T cell recognition to self epitope presented on the antigen presenting cell rather than recognizing epitope. This happens predominantly in the chronic inflammatory conditions because of repeated and prolonged inflammation both by the microbial proteins and associated tissue destruction. During the prolonged period of inflammation, the cells may shift the recognition from microbial proteins to self or tissue proteins. Another is non-specific activation by the T helper cells. This leads to cytokine influx and localized cell death. Look at this picture. This picture shows some of the autoimmune diseases which has been classified as based on the sex. If you look at this, some of the autoimmune diseases are more in females, some are almost equivalent in males and females, and some are more common in males. So, some of the diseases will have more gender predilection, either male or female, and some may not. Now, let us look at the common autoimmune diseases. First type of autoimmune diseases are like autoimmune hemolytic anemia, in which the self antigen is present on RBC membranes. This will lead to autoantibodies acting on the RBCs and lead to destruction of RBCs causing hemolysis. Similarly, it could be either drug induced or it could be because against the intrinsic factor in the stomach which leads to pernicious anemia. If it acts against the platelet membrane protein, it will lead to destruction of platelets. It is called idiopathic thrombocytopenic purpura. Similarly, in a syndrome called Goodpasture syndrome, autoantibodies will bind against the basement membrane cells of the kidney and lungs, leading to damage to both kidneys and lungs. In myasthenia gravis, autoantibodies are produced against the acetylcholine receptor. Thus, these receptors will be blocked and neurotransmission will be altered. Similarly, in Graves' disease, autoantibody is produced against the thyroid stimulating hormone. Now, the thyroid stimulating hormone will lead, if antibodies are produced, will lead to a hyperthyroid state. Similarly, Hashimoto thyroiditis or even post-streptococcal glomerulonephritis. Now, keep in mind, post-streptococcal glomerulonephritis is a very good example of molecular mimicry. After a streptococcal sore throat infection or a wound infection, cutaneous infection, the antigen sharing occurs which leads to damage to the kidneys. Other autoimmune disease would include SLE, in which autoantibodies are produced against the DNA nuclear protein, or rheumatoid arthritis, where the autoantibodies are produced against the FC region of the immunoglobulin G. Yogen syndrome, in which ribonuclear protein like SSA and SSB. Scleroderma, seronegative spondyloarthropathies, multiple sclerosis, in which the antibodies are produced against brain, mainly activated by the T cells. So, the neural damage occurs in multiple sclerosis. So, this is a very small gist of the autoimmune diseases. There are many autoimmune diseases, and more types of autoimmune diseases are being discovered even now. Coming to the laboratory diagnosis, how can we diagnose them? Like in autoimmune hemolytic anemia, this is mainly mediated by the antibodies. So, we can detect by Coombs test or directly detecting the IgGs. But like in Goodpasture syndrome, the IgGs are deposited against on the kidneys and the lungs. For detection of that, we need to do a biopsy and show the presence of these antibodies. In SLE, we can diagnose by detecting the presence of the anti-nuclear antibody and anti-DNS. Similar to SLE, in scleroderma, anti-scleroderma 70, and in Yogen syndrome, anti-SSA or anti-Rho, anti-La antibodies can be demonstrated. In rheumatoid, we can detect the RA factor, anti-citrulline peptide. And in many of the diseases, especially mediated by the T cell immune response, the antibody detection will not help, rather biopsies will evident the presence of the cytotoxic activity of the cells, thereby leading to the diagnosis. Now, having understood till this part, very simply I will address the management of autoantibodies. The basic problem is with erratic immune response. Earlier, the management was predominantly directed to suppression of immune system. Like if you give steroid, the immune suppression will occur. Thus, the patient outcome increases. But also keep in mind, when we do suppress, what will happen? The normal functions of immune system will also be suppressed. So, these patients will now become more prone to infections. Even the immune system will slowly fail to act. So, for proper management of autoimmune disease, what we do need is not the generalized suppression of the immune system, but rather specific suppression of the part by which the triggering occurs. That means, if we look at this diagram, if we give immune suppressive therapy, all parts of the immune system will be suppressed. But what we do need is suppression of the improperly acting parts, so that the patient do have a normal immune status to prevent infections further on. This is mediated by specific immunotherapy targeted against the specific antigens, antibodies, or even directed towards specific cells. Okay. There are many types of monoclonal antibodies that have been developed, which targets either cytokines so that the activation can be stopped, or it will attack particular cells which triggers the immune response. That would conclude this class about autoimmune disease. So, to summarize, we have the immune system. If it works normally, it's fine. If it doesn't work because of deficiency, we have immune deficiency. And if it works in an altered way, we can have two types. Either hyperactivity of the immune system called hypersensitivity, or it can act against the self body's own organs, which call it as autoimmune system. This autoimmune disease can be, or action can be mediated both by the T cell and the B cell, which will lead to either direct cytotoxicity or production of auto antibodies. Diagnosis will then depend on whether it is mediated by antibodies or direct cell toxicity. Come to the management. Previously, the management was generalized immune suppression. Nowadays, we can do targeted therapy to target the specific wound where the alteration occurs.